Continuing with photosynthesis, this is section 8.5, which covers the light-dependent reactions. The learning outcomes for this section are 1, to compare the function of the two photosystems in green plants, and 2, to explain how light reactions generate ATP and NADPH. So if we look at the basic processes that occur during the light-dependent reactions, we have 4 here. The first 2 are part of what we can refer to as the capture of light energy. And so the first process would be the primary photo event. Here, a photon of light of certain wavelengths is going to be captured by the pigment molecules. This is going to create a charge separation, and here the energy is going to be transferred. That was captured by the pigment will be transferred to the chlorophyll of the reaction center, and the excited electron is going to be transferred to an acceptor molecule. Okay. Following that, the acceptor molecule is going to transfer to other molecules in an electron transport chain. The electrons eventually end up being used to reduce a molecule of NADP+, which is similar to NAD in cellular respiration. And then number 4, chemiosmosis, here where we're going to use a proton gradient that's generated from the electron transport. And that proton gradient, the energy of it is going to be used to produce ATP in a similar way that we saw in the mitochondria. So first we're going to look at what's called a cyclic photophosphorylation. And here the cyclic refers to the fact that the electron that is taken away from our reaction center chlorophyll actually comes back to the chlorophyll instead of being used to reduce NADP+. For example, in sulfur bacteria, there's only one photosystem there, and it's going to generate ATP by electron transport, and this is an anoxygenic photosynthesis, so no oxygen is produced here, and the reason for that is that the electron returns back to the chlorophyll molecule. In the case of green plants, we're going to see in a bit, the reason why we have oxygen produced is because the electron gets carried away completely and then used to reduce another molecule, so the chlorophyll and the complex there has a void, and this void creates a great oxidation power and helps to oxidize water molecules. So there we split water molecule, which generates O2, but that's in green plants. So here we're not having that. This is anoxygenic photosynthesis. So here the excited electron is going to pass through electron transport chain, and then it's going to be used to generate this proton gradient or build up of protons across the membrane that, when let back through an ATP synthase molecule or ATP synthase complex, that ATP synthase can be used to generate ATP. So you can see here in the diagram on the right, here's your photon of light coming in. It gets passed around from one chlorophyll to another, eventually going to the reaction center. The electron is excited and energized. It is then going to be captured by an electron acceptor and then passed on to what's called a BC1 complex, which is going to be used to generate that proton gradient, which ultimately gives us ATP, and then following more of the chain, bringing the electron, bringing it back to the chlorophyll molecule. So there's the cyclical pattern here. The electron is not taken away, but brought back. For green plants and algae, the chloroplasts in there are going to have 2 photosystems instead of 1, and they're named by photosystem 1 and 2, and actually they're given these names based on their discovery or their description. So photosystem 1 was described first, then photosystem 2. They also go by the names P700 and P680 based on the wavelengths that the reaction center chlorophylls respond to best. Now it's the same chlorophyll A, but the reason they vary slightly in their optimum absorption wavelengths is based on the proteins that are part of their complex. So with variation in those proteins creates some variation in how the chlorophyll molecules absorb that energy. Again, this is in green plants, so here our excited electron is going to be carried away, so we need to replace that electron by splitting water or oxidizing water and taking electrons from it to replace the electron void when it's carried away. And the result here is it's oxygenic. It gives us oxygen in the process. Over here, photosystem 1 is going to function like sulfur bacteria, and I'm not going to explain that too much, but you can go back and think about that for a bit. But more important that you understand the basic flow of electrons here within a chloroplast. And then photosystem 2 here can, this is going to be the one that can generate enough oxidation potential to basically oxidize water. So in the diagram over here that you see on the right, it's called a Z diagram. Electrons don't really move in this Z pattern. It's just intended right here to scale the level of energy in the electron. As the electron becomes excited by our photons of light and moves to a higher energy level or higher energy. So there's where this scale is coming from. And then as it gets passed down the chain, that energy is being used to do work. For example, right here, we're going to be pumping protons into the thylakoid, building up this great concentration of protons, which then are going to be used to make ATP. So once that is done, then the electron doesn't get passed back to our photosystem, and instead comes to another photosystem, and then another photon of light at the right wavelength. In this case, at 680 nanometers, will excite this electrons, and then they get passed through another chain, and one of the protein complexes in there is NADP reductase, which is going to take our NADP and add the electron on there and proton and give us our reduced molecule NADPH. So the two photosystems, basically, if we looked at that diagram there, the two photosystems basically are going to be working together to carry out what's called non-cyclic transfer of electrons, because the electrons don't come back. Ultimately, these light-dependent reactions are going to produce ATP and NADPH, which are going to be used for the light-independent reactions, where we actually go to make reduce carbon dioxide to make glucose or organic molecules. So for photosystem 1 and 2, there's a Z diagram again on the right. Photosystem 1 is basically going to transfer electrons ultimately to NADP plus to produce NADPH. So we're going to reduce it. So that's referring to this photosystem right here. That's where those electrons coming from this photosystem are going to be attached to, along with a proton, to reduce our NAD plus to NADPH. The electrons that are going to be lost from photosystem 1, though, are going to have a void. So where do those electrons come from? They're going to come from the ones that are going to be taken from photosystem 2. Okay? So that's how those are replaced. And then photosystem 2 now is going to be missing. So this one here is going to be missing its electrons. So the way it's going to replace that is by oxidizing water to replace it. So here we're going to split water. You can see the water being split there, which will give us some protons right here and some oxygen there. The two photosystems are going to be connected by our cytochrome, or what's called the B6F complex. That's this complex of protein molecules here that are responsible for pumping protons into the thylakoid. So the non-cyclic photophosphorylation, in this non-cyclic type of phosphorylation, where we are going to take a phosphate, an inorganic phosphate, and phosphorylate it to adenosine diphosphate to make our ATP, what we've seen is how the ATP is going to be produced by increasing the concentration of protons in the thylakoid, and we're also going to end up producing an NADPH in this process. So it's non-cyclic because the electrons do not circle back to any of the chlorophyll molecules. Those electrons actually go back to actually go to reducing NADP+. So the photosystems then are going to replenish their electrons ultimately by splitting water, which generates electrons. And this process we've already seen can be explained by the Z diagram. I want to encourage you as a student to sketch this diagram, label it, and then practice explaining or writing about it. Either explain it to someone else using all the key terms like photon and electrons and the names of the photosystems and some of these carriers that are along the way. And if you do that, you'll be able to visualize this process and be able to answer all kinds of questions regarding how the energy changes in the transfer of electrons is a non-cyclic in fashion. So basically this diagram provides a nice summary of all the slides that were just covered right now. So that's a recommendation. Sketch it out, explain it. Overall right here you can see the rate of photosynthesis. So this is a diagram showing the importance of both wavelengths of light in producing photosynthesis here. So here's the rate of photosynthesis. We have lower and then higher. And then we see over here this is far red. And then here's red light on. Right? And then we're turning it off, turning it on. And what do I mean by far red? If we go back to the visible spectrum, far red is getting out to 700 nanometers or higher. You're starting to get to the point where human vision can no longer sense it. So this is a far red light, and then here we're about 680 nanometers, which remember these are the wavelengths that the photosystems 1 and 2 are capable of or exhibit their maximum absorbances of light energy. So you can see if you just expose the plants to far red light only, you get a certain amount of photosynthesis, a certain level. Then you turn off the light, turn on red light instead of far red, and you get a certain amount of photosynthesis, because remember the scale over here, right here is the rate of photosynthesis. But if you put both lights on, both far red and red light, you get a lot more photosynthesis. So this tells us the importance of both photosystems in the process of those light-dependent reactions. Overall, if we relate everything we've been talking about to the thylakoid, which is modeled in that image below on this slide here, you can see the phospholipid bilayer. Out here would be the stroma, the surrounding the thylakoid. We're still inside the chloroplast here, the stroma. This is the thylakoid membrane, and inside is the thylakoid space. Okay? The thylakoid membrane is going to be the one that has all of these protein pigment complexes and electron carriers that you see as you move along there. And if we go back and we relate to the Z diagram, this one right here, this is photosystem 2 right here, and then there's photosystem 1. This is that B6F complex that pumps protons in, and you can see those electrons moving along this chain and the photons being absorbed by both. Here is your NADP reductase right here, where NADPH is produced, and if we build up a bunch of protons here, they can't just go right through the phospholipid bilayers we discussed in chemiosmosis within the mitochondria. Same concept here. Those positively charged protons can't just walk right through the plasma membrane. Instead, they're going to go through channels of protein complexes called ATP synthase. The kinetic energy of those protons moving through there causes this part of the ATP synthase to rotate, which changes the shape of those proteins and activates the enzyme for us to generate ATP here from ADP. So that's the overall, we see that the structure of the chloroplast. So there's a certain structure to this. So going in and looking at photosystem 2 in a little bit more detail, which this is photosystem 2 right here. It resembles the reaction center of purple bacteria, which is mentioned on the prior slide. And this reaction center consists of 10 proteins, 10 transmembrane proteins, so not just chlorophyll A, we also have proteins associated with it. And it's the combination of chlorophyll A and the arrangement of those 10 proteins that allows us to absorb light best at 680 nanometers. The reaction center, though, is going to differ from purple bacteria. And so it differs from purple bacteria in that it also contains 4 manganese atoms, which are important in the oxidation of water. Recall that purple bacteria don't split water, and so no oxygen is generated from purple bacteria. We also have that B6F complex, which is right here. That's the one that pumps the protons, uses the energy of the electrons to pump protons into the thylakoid space inside the thylakoid. Then we have photosystem 1, where we have the reaction center as well in there, which consists of other kinds or also transmembrane proteins. But instead of 10, we have 12 to 14 protein subunits in there. And this particular arrangement of those protons with chlorophyll A is what allows this particular photosystem to absorb light best at certainly a slightly different wavelength of 700 nanometers instead of 680. Here, the photosystem 1 accepts electrons from plastocyanin, and here is your plastocyanin right here. That's one of those carriers within the membrane. And that's because we lose an electron. That electron, they got excited, got transferred onto NADPH over here, so we got to replace it. Where is it coming from? The electron coming from plastocyanin was taken from our photosystem 2 over here. Ultimately, these electrons don't come back. They get used to reduce NADP to NADPH. So this is that larger diagram again. And again, I might suggest, just like with the Z diagram, you sketch and label your photosystems and all the important things. The photosystems, some of the major membrane structures in there, including the B6F complex. That would be important when you're trying to explain how the proton gradient is. Now, whether or not you remember plastoquinone and plastocyanin, those are just electron carriers. They become reduced and then oxidized. When they get those electrons, they reduce and then they pass them on. They're now oxidized. So they're capable of this reversible transfer of these electrons. But the main ones to remember are going to be photosystem 2, B6F complex, which pumps the protons, photosystem 1, and then this other complex, which serves as an enzyme. We can tell because it ends in ASE. So what does it do? It reduces NADP plus to NADPH. And then, of course, remember the complex proteins, the ATP synthase, which we studied in the chapter on oxidative phosphorylation. So chemiosmosis is the same principle that we saw in mitochondria. If we are pumping the protons into the thylakoid space, you build up this huge concentration in there. This creates an electrochemical gradient, and that potential energy within that from that gradient of greater concentration of positive charge and greater concentration of hydrogen ions inside, they're going to want to flow back out. So there's that potential energy can be used to help us synthesize. So this is no different than what we saw in the chloroplast. The only difference here is in the mitochondria, we had a buildup of protons in the intermembrane space, and they went back into the matrix. Here, if we look at a thylakoid, the protons are built up inside the thylakoids, and they move out down their concentration gradient instead. But that energy is still used to create ATPs. Once all of that is outside of, in our chloroplast, outside of these thylakoids is where you have the stroma. And here, the stroma is going to have those enzymes that are going to drive those light-independent reactions where we're going to go and fix carbon fixation, which is called the Calvin cycle. And the molecules that are created during the light reactions, which is ATP and NADPH, are going to be used to they're going to be used to generate so NADPH. These are going to be used to help power with ATP and reduce CO2, so that they help power the cycle, and this molecule has some reducing power to help us reduce carbon dioxide to make our organic molecules of glucose. Overall, the process we've been describing is non-cyclic. However, this non-cyclic process actually does not produce enough ATP to reduce a molecule of carbon dioxide. If we're creating ATP and we're creating NADPH, as we've shown, there's not enough ATP in that non-cyclic process. So there is also a cyclic process that occurs between these photosystems. So we actually will short circuit one of them. So instead of the electron continuing on to reducing NADP, the electron is brought back. And so I kind of searched around for an image to illustrate this, and I found one. It's on the top right corner there. And what this does is allows us to generate a bit more ATP, but we're also, again, it doesn't cycle back continuously. It's only under certain conditions. We create more ATP this way, and we're still going to produce our NADPH. So let's zoom in a little bit over here to see in this diagram here on the top right. Here, it looks like the Z diagram we were looking at earlier. You can see right down here, I can't write when I'm zoomed in here, but my little hand moving around, you can follow. There's your photosystem 2, over here's photosystem 1. So again, photon of light is going to come in here at it's a P680 or at 680 nanometers, and we get our electron excited, gets captured, goes through our electron transport, goes through that complex, that B6F complex, which pumps protons into the thylakoid for we can produce ATP. Then the electron comes to photosystem 1 and then gets excited again with the correct wavelength of light. Instead of going down to the NADP reductase to go and reduce NADP to make NADPH, instead that the primary acceptor, which is the FD, the ferredoxin, is going to, instead of passing it on to the reductase, brings it back to that B6F complex, which that energized electron can then be used to pump more ATP. So here we have our short circuit going on right there. So again, this will happen under certain conditions when we need more ATP to go help drive those so-called dark reactions of the Calvin cycle.